Okay, well, welcome everyone. My name is Dr. Raven Baxter. I'm Director of Science Communication at the Cohen Center for Recovery from Complex Chronic Illness. And here I have today with me Dr. David Putrino, who's the NASH Family Director of CORE, and also Dr. Amy Proal, who is the Scientific Director of CORE and also head of the Polybio Research Foundation. And we have organized a series of monthly live educational talks where we talk about different drivers and roots of complex chronic illness and invite the audience to learn along with us and from us and share what they know, and also have an opportunity to ask questions about some of the things that are relevant to our discussion today. So if you are watching, we already have about 50 people here watching us. So if you are here, make sure you say hi. Make sure that you know how to enter in a comment in the chat. We want to hear from you. We want this to be engaging and give you all our opportunity to participate. So thank you for watching. Okay, so today's topic is on microbiome imbalance. And we had a fun illustration that went out on Twitter, actually. And it was a wonderful illustration that we worked on with Matteo Farinella, who's a very talented medical and scientific illustrator that illustrated it's a cartoon, but essentially, there is a balance of bacteria, microbes in our gut, in our body, on our body, that help us to basically carry out our day-to-day functioning. I think when you think of germs, you might think of disease, but the reality is that microbes, which are essentially germs, do other things too, right? They can help us be healthy. They can also generate illness, but it also depends on the state, that the body is in. So we're going to get into all of that today. And I think that we're going to learn a lot together. So Amy is our resident enthusiast expert, just all knowing, just kidding. There's always more to know, but it is always astounding how much I learned that Amy does know about the microbiome. But keep things simple. Amy, let's talk about the definition of the microbiome itself.
Sure. So I think it's really straightforward. It's all the microbes, all the organisms in and on us. And then when we're talking about a biome, it refers to a community or alludes to an ecosystem, right? And what's important to understand is that there are many of these organisms in and on us. There are trillions, right? And so they're different types of organisms. They're bacterial organisms, they're fungal organisms, they're archaea, which are sort of a type of, they're a little bit similar to bacteria, but different. And then there are even viral components of these ecosystems called bacteriophage, which are viruses that infect the bacteria in the ecosystems. And so what you really have is the human body is seeded with these ecosystems of organisms, and those are collectively known as the human microbiome. And sometimes that bacteriophage component is known on its own as the human virome. But really, we can just talk about them all together. And the main thing to understand is that humans are not sterile. In other words, we for a long time, and honestly, some parts of medical education still don't really bring this into current medical education. Humans were thought to just not have organisms in and on us. It was thought that the diseases that we have basically had to be a product of some kind of our own faulty human genes. And this has been, over the past decades, there's been this explosion in knowledge, and it really tied into new tools and technologies that have allowed us to identify many more organisms in the human body over time much more reliably, that we have these vast ecosystems in us that are almost equivalent to human rainforest communities in terms of like their ecosystem, their diversity and the interactions that they have with each other. And so if you think about it that way, what humans really are in a sense, some people use the word superorganism to describe humans because the collective product of what we do every day as humans is controlled by our human genes, but also by the genes and the proteins created by these trillions of microbial inhabitants. And some people sometimes use the word holobiont, and similar to superorganism, to describe that. In other words, we have, we're not just human, we're human and microbial combined together. And I think that why I'm such a huge nerd for the microbiome is once I started to understand that early on in my scientific training, it had a huge impact on how I started to just think about what it meant to be human. And so when you're studying human chronic disease, you then have to factor the microbiome into most parts and pictures of chronic disease, at least give or take, or you're likely missing potential contributors or parts of the process.
Yes, thank you, Amy. And so just to give people an idea of the scale of how microbial are we, what is the ratio roughly between humans and people? Well, not humans.
Yeah, and the organisms.
Yes.
The current thinking is that we're about, like for all our human genes, maybe we have about the same number of microbial genes. And so it's about a 1-to-1 ratio, maybe a little bit more microbial than human. So potentially we're outnumbered by our human genes, are outnumbered by our microbial genes. And that's the bacterial component of the microbiome. When you bring in those bacteriophages, the thinking right now is that there are 10 times as many bacteriophage for any given bacteria that's part of the microbiome community. So really, there are, in a sense, trillions of potential bacteriophage. So like the phage numbers get pretty crazy pretty quickly. There's teams that go into the trillions when it comes to those viruses that infect bacteria. But just going to the bacterial, the microbial genomes, we're about at 1-to-1 with them, maybe a little bit more on the microbial side.
So we have viruses, 1-to-1 bacterial to human cells.
Pretty much, yeah.
In the human body, pretty much. And then on top of that, for bacteria, there are, did you say billions or trillions?
Yeah, I mean, because there are millions already of the bacteria, and there's probably 10 times more of the bacteriophage than there are of the bacteria. If you start to add those numbers, it goes into the trillions for the bacteriophage.
So there are trillions of viruses that impact bacteria.
Yeah.
And they alter bacterial behavior and modulate bacterial behavior.
Yeah.
Bacterial behavior modulates our health and wellness. So there's a lot going on here. And it's very interesting. I want to hear your answer to how much we actually know about what's going on.
No, I mean, it's still in its infancy, what we really know. I think we've made a huge amount of progress in terms of understanding the microbiome. So we're able to, literally about in the 1980s, we thought that, science thought that there were maybe about 25 bacteria that infected and maybe could persist in humans. And the reason for that was because when you were looking for bacteria in the human body, they would use these culture-based methods. So if you could picture a scientist, like a stereotypical person in a white lab coat, like in the lab with a petri dish, that's kind of like trying to grow bacteria in that dish. That was the way that people were looking for bacteria in the human body. And what happened is that around the year 2000, when we started to use computer-based technologies to sequence the human genome, and there was this explosion in understanding our human genes, that's when the Human Genome Project, that was like a almost competition between Craig Venner and the NIH and Francis Collins. I don't know if people, that's a story for another day. When the human genome was sequenced, people said, let's take those computer-based tools that can identify genetic material and turn those onto organism genomes. In other words, trying to find the backbones, the DNA or the RNA backbones of bacteria or the organisms in the body with these computer-based tools. And when they did that, the samples just exploded with like new organisms showing up that had never previously been even understood to exist. So one of the first courses, for example, that I took when I was getting a PhD was at the J. Craig Venter Institute, where they were doing these early courses and using computer tools to find organisms that might be in the human microbiome. And in the class that I took, we discovered several new organisms on the first day. So like what that did was just humble me already, then to be like, yeah, there's a lot we're still learning, right? And it hasn't changed. There's now so many more analysis that have uncovered all these bacteria in different body sites. And now we understand a lot more of the fact that these bacteria are producing compounds and proteins as part of their lifestyles. We understand that a lot of those compounds interact with our human signaling pathways and impact for everything from our hormonal signaling to our mitochondrial metabolism to all kinds of human processes. But the truth is that we still are learning every day about how these organisms contribute to health and disease. And on the virome side of those bacteriophage, it's really early on in the game. There are still teams that call the bacteriophage in the human body dark matter. It's really, we know about as much about these phage in the human body as we do about the solar system. Like we know there's stars out there. We know that like we've named some, but the truth is that overall, we just need a lot more exploration. So the phage are, kind of an open question. Yeah. In fact, if you work on phage, you can just, it's one of my goals. One would be named after me. Like, they cover new ones all the time. So one of the people I work with, he has a couple of phage named after him. And, there's plenty of opportunity. There's trillions if you get into it.
I love it. It's like when people name, to your point, new stars. There's
exactly.
tons of stars out there.
Yeah.
Somebody sent me a star named after me, and like it's just
that's cool
coordinates, and I'll never be able to find it. But like,
hey, cool.
I hope that you get your phage named after you. What will you name it? Like the Amy?
Yeah, that's the question, right? I don't know. Maybe something more with Proal, my last name. I don't know. I gotta think about it more.
And I hope it's like a fun-shaped one that we've never seen. I think T4 bacteriophages sounds so nerdy. I think T4 bacteriophages look really cool.
They are cool. There's, we got to show, like, just Google bacteriophage to anyone watching this right now. You have this, it looks like an alien too. It's like a kind of capsid, and then they have legs. That's what they use when they infect a bacteria, right? They kind of attach to it, and they use that to break the bacteria up and disturb its replication, its growth. But they look like little aliens as they do it. So we went through a period with Polybio where we actually 3D printed phage. We had these quick, cool phage keychains. Like, no, they're really cool. I mean, yeah, but so yeah, and they're in us as we speak.
Woo!
Yeah.
I mean, we kind of are like bionic. I don't know.
Yeah.
Well, that's a stretch, but the girl can dream. So I think, we touched on the things that we don't know. Let's touch on some of the things that we do know. One of the things that I learned about bacteria that I found fascinating when I got started learning about microbiology back in college is that they help us synthesize vitamins.
Yeah.
I thought that was mind-blowing. Who would have thought that something that, like the media, every now and then we hear about, E. coli breakout, Listeria breakout, food recall, a lot of the things that we hear out in the news or in social media, when we hear bacteria, we think, oh no, bad, bad, bad.
Mm-hmm.
They stay away. But reality is bacteria actually help us synthesize things that we can use to survive. Can we talk more about the more commonly known ways that bacteria impact our everyday lives?
Yeah, I think so. Obviously, it's it's like a rainforest or a forest. You can have a lot of animals in the rainforest interacting together, and everything's, yes, there are relationships where animals are eating each other and things, but if it's in a state of balance or homeostasis, then most things are going okay, right? It's the same with the microbiome. Someone who is totally healthy, or an Olympic athlete, or someone who's functioning at a high level, has all these ecosystems in them. So obviously you can be healthy and you have a microbiome. And under those conditions of health, the organisms in and on us produce a lot of different compounds. So some do produce vitamins. They also produce a lot of amino acids. So for example, there's certain bacteria in our guts or other body sites potentially that produce tryptophan, for example, which is an amino acid. And tryptophan then, is a precursor for serotonin, which is a neurotransmitter. So that can impact, signaling of how our nervous system works. Also, there are certain organisms, for example, that have been shown to produce or modulate, for example, how GABA, which is another neurotransmitter, is produced or broken down or metabolized. So we have this definite thing where organisms are contributing to the compounds that are circulating throughout us, and that are parts of the signaling pathways that, like the metabolites they're making, are part of the pathways that signal for things to happen in humans that allow us to think or process things, right? Or digest, or you name it, like all kinds of processes. Now, our human genes usually code for the same thing. So for example, our human genes alone can make tryptophan. So it's not like, in many instances, there's sort of a redundancy where we make it as humans, but our microbes also make it too. So there's like an interesting relationship there where you have this sort of mix of the human produced, let's say, tryptophan, the microbial produced tryptophan, and the combination of those, amounts of tryptophan is kind of what's, leading to a certain person's levels or signaling capacity at a certain point in time, right? So that's a big part of what microbiome does. And, I think what's important is that even when the organisms, let's say, are in the gut, the metabolites they make sometimes end up in the blood. There's actually a study, I think it was almost in 2009, by a group, Gary Sudzak's lab at Scripps. And it really was also important in my thinking about microbiome, because he showed that the majority of metabolites that their team found in the blood were not human. They were actually produced by the gut microbiome and had gotten into the blood. And so we can't even make this assumption that the metabolites that are circulating in us, even in our blood, which has a lower microbial burden technically in healthy people, the metabolites or the products made by these organisms still circulate. So really, they can get to almost any part of the human body where they could theoretically interact with a lot of our signaling pathways.
And
Yes.
I just want to point
Go
out
ahead, David.
that, what's really, some of the stuff that's really, really cool about this is that, downstream that can lead to behavioral change, So, if you have more serotonin available to you, or if you have more dopamine available to you, that can change how your mind and your, and your body works. And there's all sorts of really cool studies that show that, like, when you put someone on a diet that makes something like tyrosine, which is a precursor of dopamine, more available to them, they will display more fairness in, choosing to accept or reject monetary payoffs. There are studies that show this. Your social decision-making changes as a result of your gut microbiome being altered. And so, there's a lot of, incredibly important nuance that goes along with what could be happening in somebody's cognitive, social, and mental health that can be linked to a very, concrete issue with gut dysbiosis. And these are some of the sort of, I think, very complex problems that we need to sort of start to disentangle with good science.
Yeah, and that goes in, like, one of the the bigger topics in microbiome is the gut-brain axis, which really is what David's talking about, to a point where the site of the body that has the most of these microbial ecosystems is the gut. And the gut signals those organisms, whether it's the products or the metabolites, anything they're doing, signal via vagus nerve or via other pathways directly to the brain. And so what the microbes are doing and signaling there can just have this direct route to what happens in the brain. And that's just one mechanism. And thus, when people say like, oh, I have a gut feeling about something, or like these like sayings are out there, there actually is biology behind that kind of idea, which is that if someone has changes in their thinking, or even some of the disorders or conditions that involve, whether it's neurological or neuropsychiatric symptoms, well, organisms could be involved in those changes, and that's something that medicine doesn't think so hard through right now, that could be pretty impactful if we start to look at it more closely.
I'm tempted to go straight to the gut and talk about all of the different gut axes. But where are some other places that we should pay attention to when we're talking about microbial imbalance?
Yeah, so the thing too, and when you say imbalance, is that the thing with the microbiome is that, as I said before, under conditions of health, especially when the immune system is in good shape, then you have these ecosystems that are functioning, cooperatively, if you will. But then under conditions of immune dysfunction, especially, or imbalance or immunosuppression, you can have what's called dysbiosis, where a community of organisms, whether it's in the gut or another body site, for example, the lungs have a low-level microbiome, the mouth has a really robust microbiome community, they shift, the organisms collectively shift, like as a group, towards a state of imbalance, where now the products and the signaling that they're doing technically can throw off or are not signaling well. And then that could throw off the balance of whatever they're impacting, which is, as we've discussed, many things, right? So this dysbiosis, which is associated with many different chronic disease states now, from Crohn's disease to ulcerative colitis to even diabetes. So many conditions are increasingly tied to a place where these ecosystems go out of whack, if you will. And I think, and that can happen in a number of body sites. And the locations in which microbiome communities are the most robust are gut, also, like I said, mouth, and then to a degree, there are communities in the lungs. It's a low biomass is the word that's used when it's like a low number of organisms. Technically, like some people think the lung microbiome turns over pretty often. In other words, that how stable it is still a question. That's the case for organisms in other body sites too, is the gut and the mouth definitely seem to have these communities that are stable no matter what. But in other body sites, we know that they're not sterile anymore, but the degree to which, let's say in the liver or the kidneys, or even, will sometimes find microbes there. And it could be a community, but it might depend on the person, their immune status, health. It could depend on how many organisms have sort of translocated, which is sort of shifted through barriers into that site. So the main thinking is that we're not sterile in too many body sites, including, honestly, even in blood in a lot of people. It depends. But the most robust ecosystems would be the gut, mouth, and actually even vaginal microbiome in women is huge. I mean, there's a really robust vaginal microbiome that's studied in all kinds of capacities, including impact on sexually transmitted diseases. If the vaginal microbiome is imbalanced, for example, women are more likely to acquire HIV than if it's in good shape. So you can have a direct impact on how other pathogens are able to colonize. And there are debates on if a baby born via C-section, for example, would not doesn't pass through the vaginal canal, and then thus isn't sort of seeded with vaginal organisms as some babies are who are born naturally. So there's questions about vaginal microbiome and birth and the seeding of babies. So that's another community that has a lot of considerations to it, is the vaginal microbiome, because it affects how infants start out in the womb, or how they start out when they're early in life, I would say. And there's even been the womb for a while was thought to have microbiome. There's about 50, it's a 50-50. About 50% of research teams have kind of found communities of organisms in the womb that would be classified as microbiome, the womb itself, while other teams argue that those might be there only under certain conditions. So there's still kind of a debate on whether the womb microbiome is totally there. That being said, like, groups of organisms that get into the womb can have an impact on the developing infant. That's just not contested. So whether you want to call it a microbiome community or not, like, the impact of organisms collectively in the womb is a major consideration for the beginning of a human's life and how they're set up for the rest of their life.
It's really important. I want to go back to the mouth because I feel like that's a really accessible place for most people. And bacterial plaques on our teeth that's dental plaque, right? And one of the coolest things that I learned in school that I think people watching might find fascinating is that like, they are communities of bacteria and they talk to each other, right? Through quorum sensing. And that communication is essential for formation of dental plaque. They produce things called autoinducers, right? So
yeah, I mean, I think, quorum sensing is the best term. And by the way, we all already agreed that I'm mostly going to talk on this one, David. David, chime in anytime, but I
knew once I went down quorum sensing, we were going to
exactly. There's no way that I'm not answering this one too. So I'll just, David, you can keep typing. What are we doing? Yeah. But yeah, because it really is. So, we floss our teeth. Why are you doing that? Well, what you're breaking up, that plaque, are biofilm. And what biofilm are is you have bacterial organisms that get together, and this is really obvious and intelligent. One bacteria alone in, let's say, like the mouth, isn't going to stand much of a chance of survival because the immune system, all the immune cells are going to see it and target it and kill it. So if it can team up with other bacteria, and again, these are others in its microbiome community, right? It stands a better chance of survival. So what bacteria do as they shift towards that more dysbiosis problematic state is that they often form biofilm, which are groups of bacteria, and they embed themselves in a fibrin matrix, interestingly, where they're caught up inside fibrin, which makes it a lot harder for them to be broken down. But the way that they build this community, and these communities, biofilm, have even in terms of their intricacy, in some cases, been compared to cities, like how elegantly they are built. And they could be a single species that builds a community with others of its own species only, or they're even mixed biofilms where different organisms, team up together. And then because each organism in the biofilm has its own sort of strongest function, they'll even split the jobs. So let's say one is really good at defense, they put those on the outside, right? And what they're like making peptides that fight off the immune system. Whereas the one on the insides might be good at producing nutrients. So they're there and they're making the nutrients that are feeding the rest of the community. So they split roles. And the way that they do that is they have their own chemical language called quorum sensing. And they produce these signaling molecules where, depending on the concentration of the molecule, they're able to basically use that as a chemical language to know when to activate, when to get together, how to divide function. This is another part. We need to study this more. I mean, there's so much, I'll be honest, potential to disrupt biofilm if we better understand the chemical language, these quorum sensing signaling by which they're built. And there are not enough labs working on that. There's only a few that study these bacterial chemical languages, so there could be way more. But that happens, and for example, it's just as a straight up example, periodontitis, which is tooth decay. Yes, like if you have a cavity, especially that moves into the gum, what happens there is that a bacterial organism in the mouth that's usually a bad one, like P. gingivalis is one of the oral microbes that's just no good. It's always a problem player. It will signal via quorum sensing to its neighbors and say, hey, like, you guys want to form a cavity? Let's do it. Let's team up. it lets split up function in these ingenious ways where they actually, some of the organisms that start to signal with it will grow flagella. So they were, more ball shaped before, but they actually start to grow tails as part of this. So now they can better burrow into the gums, and they just take on their new forms so that they're better entering. And what is really interesting in that case is that P. gingivalis can be just 1% or less, like 0.5% of the organisms in the biofilm. It's composite, it's the number of P. gingivalis in there are small, but its signaling is so profound that it's directing the whole biofilm, right? So you have these really, they're instigators, if you will. And so, technically these bacterial instigators are a lot of how this dysbiosis happens, where one organism that's pathogenic or starting to act more pathogenic sort of starts to signal in a way that causes something detrimental and moves, and the rest of them around it get caught up in this and start to move that way too, right? And so, that's part of how a cavity forms. Yeah.
Okay. So now we get to do the exciting, or I guess interesting rather, connection back to complex chronic illness. And maybe this one isn't necessarily as complex as the other illnesses that we've talked about, but heart disease. So there is a link between oral health and heart disease, and that ties into bacteria from dental plaques entering the bloodstream, especially in people who might already have compromised gum lines, like gum disease, attaching and create or integrating themselves into heart plaques, arterial plaques. So that is a really wild connection, and I think that many more people should be aware that your mouth health, your oral health can affect your heart health. So shout out to Marcus, who's in the comments saying, I'm going to brush my teeth and floss. I think that's excellent. I am going to make sure that I floss more often. I'm going to admit I sometimes lack on the flossing, but brushing I'm good on. But got to take care of our bodies in all of the different ways that we might not expect. It's all interconnected, whether we know it or not. So,
right. Yeah, no, I like flossing because my partner and I joke, it's kind of good that you can actually access the biofilm in your teeth and break them up. I wish I had these urges to floss, my arterial plaque or something, you know what I mean? But you can't, right? So you can't floss a biofilm in your, kidney or something, right? But you can in your mouth. So it is a strength. And I think that oral health is extremely important. I was talking just now, actually, as part of a podcast to one of my colleagues that works in Alzheimer's disease, Rudy Tanzi at Harvard, and he was telling me, and a couple of groups have found this, that P. gingivalis, that instigator that I mentioned in the plaque, they find it in the Alzheimer's brain as a seeding pathogen that sometimes might get there and impact plaque formation in Alzheimer's, right? So you have these possible contributions of these bacterial organisms that can get maybe from the mouth into the brain. There's routes for that, right? Or can leak from the mouth. This was actually and is a thing with COVID. In COVID, there's teams that have found oral organisms maybe get into the lungs. And that's been an issue where I think, were studies that showed with people with periodontitis, again, gum disease, were maybe 3.5, 4, I forget the exact number, more likely to have a severe case of COVID or be hospitalized than people without periodontitis, potentially just because they already have these oral organisms that can leak into lung and cause so many more, other bacterial issues already in the lung. So, you have many, many connections between oral organisms that go wrong in chronic disease.
It's mind-blowing, and I can't help but to react to every last thing that you said, because I feel like with every sentence, it was just whoa, you just said that someone found that our oral health might play a role in our brain health.
Yeah.
Whoa. If anybody is watching this and is mind blown, please write this in the comments. I know I'm not alone here. And again, like, I think people think that all scientists know everything, but I think better uh, the better label is that we all know like what we know. And so when we get to talk to each other like this and learn what each other knows, it's always fascinating. There's always something new and interesting. So let's talk about complex chronic illness and the roles that we understand microbes play in the development, the manifestation, and I mean, even have we considered treatment pathways in consideration of the microbiome in the context of complex chronic illness?
Yeah, and David, maybe you could mention, I think there's one clinical trial of a probiotic that was encouraging in long COVID, but I think as one trend, and we've mentioned this in the pathogen reactivation session, when we're talking about, let's say, long COVID or ME/CFS, one thing that can happen is that if you get a new infection, let's say SARS-CoV-2, you get COVID, that pathogen knocks down parts of the immune system as part of how it drives disease. So, for example, interferon signaling, really important molecules that control other infections, including collective type infections, right? So just as a new infection might cause sort of like a viral reactivation of something like Epstein-Barr virus, the same trend can happen except for organisms in a collective state. Like a new infection can sink the immune system, and that could let organisms that are in a community, it's somewhere like the gut or the mouth or even the lungs, to collectively together gives them this opportunity to move to a state of imbalance that could be more problematic. So that's definitely something that we need to consider in long COVID, is that each new infection or SARS-CoV-2 infection could be an instigator, especially maybe if someone's microbiome was already, not in a great place, but it was like on edge there. And then now we gave an opportunity for it to sort of evolve in a direction where it becomes more problematic. And I think that's an issue potentially with long COVID. So we have collaborators at the J. Craig Venner Institute who are studying the bronchial lavage fluid of patients with long COVID as part of a study. We have teams looking at the oral microbiome in long COVID also to just try to understand if it's off in people and how. But there are one or two groups. There was a study, David, that probiotic study in Hong Kong? That one was encouraging.
Primo one.
Yeah, exactly. Was a good proof of concept trial that was a randomized trial that showed that people with long COVID given a probiotic that was basically, I think, Bifidobacteria lactobacillus, I hope that's right, which are two organisms that can produce compounds that sometimes are more beneficial, seem to provide, again, did not cure people, but seem to provide some benefit just with symptoms. So that's reasonable to think about. And there's one or two other probiotics that are on our radar at CORE that we're considering, trying to either trial or give patients in a reasonable capacity to see if, we can bring back a little bit of the health of the gut or something in a way if it's been thrown off by an infection like SARS-CoV-2.
Yeah, I like the analogy that we've been floating around with in terms of the rainforest, or even if you're curating your microbiome, the garden,
Exactly.
If you don't take care of the garden, the weeds come in and overrun the plants that you're trying to curate. If you're not curating your microbiome, if you've never even thought about your microbiome and you've got a rainforest, if conditions change, invasive species can come in and and cause all sorts of problems. And I think that right now when we think about how the microbiome can improve health, it may not always be the case that fixing this gut dysbiosis is going to fix all of your problems, because again, we may not be dealing with that initial underlying cause that caused the dysbiosis to begin with. But if you can correct the dysbiosis, then as we've discussed, things like serotonin or dopamine or vitamin metabolism or nutrient metabolism that your body was not able to do before, or was having a difficult time doing before is now becoming much, much easier. So your body gets the hormones it needs, the body gets the neurotransmitters it needs, gets the nutrients that it needs. On the other hand, you can also curate your microbiome to take bad things out of the system, and so one of the things that we're looking at right now is a probiotic that may be able to take lactic acid out of the system of people with long COVID, and a bunch of people with long COVID, I think, are very aware of the fact that there's often more lactic acid is seen in the blood of people with long COVID than in healthy controls. We think lactic acid is a waste product related to exertion, so this may be an indicator of PEM. So if you can encourage bacterial growth in your gut for something that metabolizes lactic acid at a much accelerated rate, of course there is a lot of promise to that. So it's not just about letting good things in, it's about and producing good things, it's about taking bad things out. That becomes important. But one of the things I want to throw right back to Amy to get Amy talking again is I think in addition to gut dysbiosis, we should also talk about leaky gut. And the etiology of leaky gut, and why, if you have leaky gut, no matter what you do for your microbiome, you're probably not going to see benefit. And in fact, that's probably, in terms of order of operations, it's probably something that needs to be addressed first if it is present.
Yeah, that's a good point, David. And that is because all of these ecosystems, especially in the gut, where there's just trillions of organisms, and even in the mouth, you have these barriers, if you will, like that keep the organisms there in the gut. So the gut lining, is important then, because what it's preventing is the leakage of organisms from the gut into the blood. And you really don't want too many organisms to leak into the blood, because if an organism gets into the blood, it can just set off a huge amount of inflammatory or other even clotting or coagulation issues. In fact, one of the ways that I became interested in Riccio Pretorius's work, which is, I think many of you know her for the discovery of the long COVID microclots, right? Is because before COVID, we're already friends, because she was showing how bacterial products, especially LPS, which is a product of many of the cell walls of bacteria, if those leak into the gut, they can set off actually clotting, similar to what she's seen in the microclots, neutrophil activation, and net formation, these big projections of the neutrophil immune cells, and then almost already a feed-forward system that leads to inflammation and potentially even impacts the blood vessels, right? So you just have this cascade of events when a bacteria or even a bacterial product enters the bloodstream. So you want to keep the organisms in the gut or the mouth, like in those sites. What happens though is if, let's say, the gut microbiome shifts to be dysbiotic or imbalanced, that will lead to generally more inflammation. In simple terms, there's going to be more problems. That's going to lead to more inflammatory products being produced by those organisms, or even the immune system, as it tries to control the problem, will create inflammation to try to address it. And that inflammation can wear down the lining of the gut. And that's what's called leaky gut. I mean, I think actually a lot of people have heard that term. And if that happens, you start to have this issue with bacterial themselves or their products leaking into blood, and that can definitely cause a lot of symptoms. So before people take probiotics, it's important to think about addressing potential leaky gut first, because otherwise, if you take a probiotic, it may get into your gut, but those organisms might just leak into the blood because the gut's leaky. And then even though they could be beneficial, they were supposed to be in the gut. Look, once stuff gets into the blood, it's not going to help out there. So there's protocols and some supplements and even some things where the first thing people work on sometimes is improving the gut lining and strengthening the lining of the gut. And then sort of as a secondary thing to take probiotics or other approaches to improve microbiome via consumption of new organisms. And luckily there are teams that are working on new therapeutics to improve gut permeability One is our colleagues at Harvard Medical School, Leo Yonker, Alessio Fasano, their team. Alessio, who's great, he's part of our long COVID consortium. He actually discovered zonulin, which is a marker of gut permeability. It's produced when the barriers of the gut lining are disrupted. And they can measure that, which we have in a couple studies looking at as a kind of sign of gut permeability. But they've also, there's a drug called larazotide that can potentially sort of help repair the junctions of the leaky gut. And they're trialing that in children with long COVID now, which is cool. So as we get an idea, for example, from that clinical trial, we can just keep, building the tools that we have to prevent leaky gut, which is, again, just part of the bigger picture of trying to think about the way we want to move as we address potential microbiome issues in most chronic conditions that are tied to dysbiosis.
I think what you said is all of it's very important, but especially because from the things that I've seen in the work that I've done at CORE, I see people who may claim to be medical professionals promote certain diets or prescriptions or supplements and kind of take advantage of this lack of knowledge about the microbiome to push a certain regimen. What do you think that people should look out for? I guess, I think there will be a subset of people here who will listen to this and be like, well, what can I do? If I can just get a probiotic from the store, what do I do? So what are your thoughts on that?
I think that one thing I just consider is that there's things that people do for their microbiome when it's in a state of health and things people do for their microbiome when it's not in a state of health and it's imbalanced. And those are two different ways of thinking. If you're healthy, for example, you'll hear a lot out there with microbiome, like you should eat a lot of fiber. You should make sure to feed your microbiome with fiber. Well, that's true if your microbiome is in a good place. So if you're healthy, a lot of a high fiber diet and foods like that could be help. You want to maintain the community that you have. It's in good shape. But that can be a problem. Let's say your microbiome's been thrown off and it's out of whack. You don't really want to feed it a bunch of fiber because you're feeding the problem. So the thing is that with people who have microbiome dysbiosis, one of the first things that they sometimes experiment with, and of course it's really individualized how things work for people, is just a more low carb diet where they're actually pulling out some fiber and they're pulling out carbohydrates, especially refined sugars, and especially just like really bigger servings of carbohydrates. So they sometimes go towards like a paleo diet where there's lower carbs. And that can help a little bit in some people's cases because bacteria feed on sugars and carbohydrates. So if you're not happy with the bacteria you have and you're giving them a bunch of sugar, carbohydrates, and fiber every meal, they're proliferating. So that move to a lower carb diet can sometimes be sort of an early step that people who are kind of experimenting with just trying to alter their microbiome is like a consideration that isn't too, otherwise crazy to try. That's one thought. There are some supplements that people use to improve gut permeability. And those are a couple out there that, I don't know, there's a book called The Gut Health Protocol that sometimes I look at and has some really basic supplements on that front. And then I think, from there, I think the other thing that people can do is think that because microbiome can be good for some people, like many organisms are really good, you want to think more intelligently about, like David was saying, that one group who's making a probiotic, for example, that can degrade lactic acid. Like it makes more sense to me, at least, to think of the mechanism of the probiotic you're taking and how it might be addressing a specific problem or filling a specific gap or issue, rather than just taking a lot of probiotics and sort of hoping for the best. Because a lot of brands of probiotics, it's almost like they're trying to outcompete each other. So they'll be like 1 billion in this pill, and then the other one's like, oh no, they made 1 billion. So they're like, okay, 2 billion in our pill, right? And there's a point where it's a little less about how many and more about what the strains are and what they're going to do. And so I think just taking a huge number of them, like you want to just, you might not need to do something like that as much either. And then I think, those would just be my main takeaways, is just be careful that just because something seems good, just to take a lot of it, that wouldn't necessarily always work.
I would say my general advice that I try and make sure is out there is, the clue is always in the title, right? Complex chronic illness. So it's complex, it's not simple. So whenever you see someone trying to oversimplify the issue, that's a red flag. So for people out there who are looking to access care, and someone is claiming that they can fix your microbiome, first of all, you want to understand what the personalized protocol is, as opposed to like, is this what you're giving to everyone? Second, it's like, okay, well, why are you giving me this protocol? What are you trying to encourage? What are you trying to discourage that's growing in my microbiome? Thirdly, and I think that this one is important, especially for microbiome interventions. Can you please give me some peer-reviewed evidence about the intervention? Because, there's a lot of there's a lot of professionals out there who will just sort of fly by the seat of their pants and say, and read a microbiome sequencing test, which can be very expensive to run, and say, well, this one's a good one and this one's a bad one, and and I'm going to sort of make you a magic potion that will get rid of the bad and improve the good. And all of that is conceptually fine, but like show us the data, because there are some really wonderful studies out there showing that if you encourage certain bacteria and if you discourage others, in certain disease states, you can improve a symptom profile. So show us, show those data and ask for those data. So, I would say that for anyone who is perhaps finding themselves staring down the barrel of some very expensive and highly specific prescriptions, the question becomes, why are you recommending this for me? What data are you basing that off of? Like, from my testing, can you show me like what's out of whack that is telling you that this is the thing for me? And then can you show me a paper that really highlights that people who have been in a similar situation to me do better?
That's interesting. I'll also add that conversely, when microbiome first started really taking off in the early 2000s, and there became a little bit of an assumption that microbiome is good, right? And it's not, and it is under certain conditions, but it can also be really problematic. It can be driving disease. And so the one thing that I also think is important is that for the treatment of some infections, especially bacterial infections, let's say if you have Lyme disease and you missed it and you have Borrelia and it hasn't cleared, you might need to be put on a protocol of antibiotics to kill your Borrelia. And I've had people come and tell me, like, I don't think I'm going to do it because those antibiotics could kill organisms in my microbiome. And since my microbiome is really good, I don't want to do it. And it's like, no. Like, it is a much bigger problem to have unaddressed Borrelia in your body than it is to potentially have your microbiome be affected, depending, as long as you're working with someone who knows what they're doing. Because one of the things is that if you have an unaddressed serious infection in your body, it is going to be always down-regulating your immune response. Like, so almost every pathogen that's unaddressed, it inhibits the immune response in order to stay in you and to drive disease. This is the same with herpes viruses sometimes, or whatever pathogen it is. And that immune dysfunction is part of what lets the microbiome get out of control. In other words, the immune system is needed to keep the bad players in the microbiome in check. So if you have an unaddressed infection like Borrelia Lyme, it's probably better to try to treat it, and then to hope that if you address that infection, your immune system might do a better job of coming back and helping you control microbiome after that point. But don't, again, like this is working with a doctor, but I wouldn't be so worried about microbiome that I would never take antibiotics under any circumstance. Some people can go really swing in a very far direction of being like afraid of antibiotics, even when they're really needed. And that's a view where you can go a little far, in my opinion, too, depending on the situation.
Thank you, Amy. And so we're wrapping up here. I do have one question for us. This person said, y'all mentioned access to biofilms in the mouth, making it easier to break them down or disrupt them. Are scientists looking at ways to disrupt them elsewhere? Wincing thinking about the brain right now. Valid.
Yeah. Lumbrokinase, which is a supplement that we're trialing at CORE in long COVID, MCFS, and patients with post-treatment or chronic Lyme disease. We're trialing it primarily for its effects on breaking up fibrin. It's an anti-fibrinolytic enzyme. But remember when I told you that what biofilm are encased in is fibrin? Lumbrokinase then is also used in some communities as a biofilm disruptor. So there are potential supplements like lumbrokinase, again, that we're doing in a trial. Again, there's got to be careful with the dosage and uses of this. That could disrupt fibrin, and by doing those things could maybe have an anti-biofilm effect. So there are people looking at that. Like if you disrupt the biofilm matrix, you've sort of released those organisms that were relying on it to stay more hidden.
Thank you. And we do have our website up now, coresinai.org, and you can look at the current research that we're doing, the clinical trials that we're running, the ones that we've completed, and the ones that we've published our results on. So if you're ever wondering what we may be up to that you can learn about, be sure to check there. Very soon, in the next few months, there will be a suite of educational resources meant for a variety of audiences that we are looking to serve. And yeah, so thank you for your question. It is 3:59. We're ending right on the dot. Thank you, everyone. We are at almost 1,000 viewers. Thank you to all 1,000 of you for coming, and we hope to see you next month. Make sure you follow us. We need 3 more followers on LinkedIn so that we can stream to LinkedIn. So please follow us on all of our platforms, but especially LinkedIn. We need you.
What's LinkedIn?
I know. I know. But we gotta get on there too. So yeah, thanks everyone.
Cool.
See you soon. Bye.
Thank you, everyone.
Bye.